There we go. It's always good to have a little extra. The Ramseys are on the way. Traffic is bad. Got it. Okay. Anyway, let's see. So I said White is here. Vance is online. Reed is online. Okay. Reedy is online. That's it for now. Does anybody have any questions while we're waiting? If we're up to me, we would go 100% online. Not because of COVID. These gas prices are getting crazy. Apparently, it's only going to get worse. I wish I lived closer to campus. Might ride a bike or run or something. Good morning, Cordone.
Good morning.
Well, since no one has questions, I might as well use this opportunity to remind some people of some things. I'm getting some questions about Google Classroom, so I might as well use this time to show you really quickly. Let's see. Let me log on here. So one of the questions I was getting was, someone was asking, a couple of people were asking for the video for the pre-lab lecture for the photosynthesis respiration lab. Because, you know, not only does it explain how to do the labs and what the labs meant, but it also explained how to do the lab report. So one of the ways you could do it, first of all, you can go through your old emails and look that way. Because I sent it, every time I post something to Google Classroom, you get a video, an email of it. So you can go through your old emails. That's one way. The other way, and this doesn't always work, I'm going to see if it does this time. If you go here to Google Classroom on the stream, which is, again, every time I post something, it goes to the stream. So I'm going to hit Control F, and let's see. Because if I hit lab, that's going to be too broad, and I'll probably get a lot of results. I don't know. Lab report. Let's see what that brings up. Here we go. And this brings up, still have not released the final grade from the photosynthesis lab. All right. So, yeah, right away when I did that, it brings this up. Here's the video, or here's the link that brings you to the photosynthesis lab. And here's the link that brings you to the photosynthesis lab report. So it's all right there. And again, all I did was hit Control F and then just typed in what I was looking for. And then, of course, you can do the organized thing, I guess, if you want to call it that, and go up here to classwork. We're still in Google Classroom, and you go up to this top middle tab called classwork, and then just look at your area. Like if you're looking for something in the lab, just scroll down here on the left for your category, which is lab. All right, and mine's going to look a little different than yours, by the way. You scroll down until you find what you're looking for. So there we go. There's the lab report. This is new. The video wasn't there when these people were asking me, but yeah, there's the video where I tell you how to write the lab report right there on the sheet that you need to write the lab report. And also, if you go down some more right here in the photosynthesis lab itself, you've got photosynthesis lab. You've got some videos about photosynthesis and respiration. You've got the PowerPoint video for my lecture. You've got me doing the lecture. And of course, you have those questions for the lecture for the people who didn't attend. So just something to think of. There's a lot of stuff on Google Classroom. If it turns out some of you probably have not looked at it, I highly recommend it. You know, look at the videos on there. There's things you can watch for independent work points, for example. If you don't like to write, I mean, I still hope you write, but you can watch videos for independent work. And I'll get into that later because now we are a few seconds away from the official start of class. So I just wanted to use that time that we had while I was recording to show you guys that. So it's 8 o'clock. Let's see. As always, if you're online, send me an email afterwards to let me know that you were here. The first attendance word is going to be chip, like a microchip or a corn chip, chip, like something you would eat. Well, of course you wouldn't eat a microchip, but you know what I'm saying. So anyway, yeah, that's the first attendance word. Make sure you send me all the attendance words by 9 a.m. Anything after that, you'll need to watch the video and give me the questions. So before we get started, even before I get started with announcements, does anybody have any questions about anything specific about Chapter 8 or just the course in general? Yes. So the question in person was, when is the lab report due? And I'm doing this on purpose. The lab report is due 2 weeks after you get your official lab grade. And I have not given anybody their official lab grade yet. I went in and graded everything like right around the due date and said, hey, make sure you get your corrections in, because next time I look at it, that'll be the end of it. So make sure you get your corrections in on the photosynthesis respiration lab report, because I still haven't given the last grade for everybody. So everybody can still fix points or fix their mistakes for full credit. And you can still come to me for help to fix your mistakes for full credit. But I'm hoping to get that done today. So yes, to go back to her question, once I give you your final grade in that same announcement or whatever, the same comment where it says, hey, your grade was blah, 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 I will also say, and your due date is this. So you will know exactly what your due date is. I will give it to you when I give you your grade for your lab. And again, it will be 2 weeks from that date. So great question. Any other questions in general or specific? All right. Well, I'll remind everybody about lab this week. I wanted it to be easy. I wanted it to be online because I know you're going to be focusing on the lab report. So if you met me online on Monday, or if you're already done and have your lab graded, then you do not need to do anything for lab this week as far as attendance. But if you do not come in on Monday and you did not finish your lab yet, then you need to meet me online for lab on Wednesday or today. So to be clear, again, we're not meeting in person, but I will be there online. And you need to be there if you have not returned your lab in and got it graded yet.
Okay, so what time do we need to meet you?
Same time. It's pretty much everything's exactly the same, other than the fact that we'll be meeting online. The same link that we're doing here. So we'll be meeting at 2 p.m.
Oh, okay.
But again, if you get it done, if you get the complete lab done and graded before then you don't even need to meet online. And I guess I should bring this up too. If anybody doesn't meet, if they didn't meet Monday and they don't meet today and they haven't gotten it done yet, then that's considered a lab absence, and then your lab will be worth 1/4 of whatever it is you earn. So be there, please. I guess that's it. That's it for my announcements. Anybody else? I'm trying to drag my feet because I know at least 2 of the students are caught in traffic, but I did what I could. So let's get into it. Let's jump back into Chapter 8. Here we go. It's been a while. Last time we talked about this was on Friday, and it is now Wednesday. Can someone please confirm that you can? I know you can hear me, so can someone please confirm that you can see the PowerPoint? All right, thank you. So yeah, to remind you what we talked about. At first, we talked about what the cell cycle accomplishes. I'm not going to get too much into that right now, but like mitosis is for asexual reproduction, meiosis is for sexual reproduction, things like that. We, as sexually reproducing organisms, do both, right? Right now, I am reproducing. Obviously, I'm not sexually reproducing right now. That would get me in a lot of trouble. I am going through asexual reproduction right now. I have dead cells that are being replaced, right? And my liver cells probably are being replaced. That's one of the very common ones. Not that you need to know that. So yeah, that's something you need to know. Then we started talking about the cell cycle of mitosis. At least that's the big bullet point. We haven't even gotten into the cell cycle of mitosis yet. First, we talked about eukaryotic chromosomes in general. And this is a good time to remind you, we humans and plants and fungi and protists, things like that, the more complicated life forms, we are eukaryotes as opposed to prokaryotes. We're not even talking about prokaryotes in this class, but you could look it up for independent work if you want. But anyway, we talked about eukaryotic chromosomes. So again, chromosomes are those things inside of our nuclei that contain all of our DNA. They're made up of about half DNA, half protein. You need to know all that. That's where all of our genes are. This is mostly what we're going to talk about this entire exam. So exam 3 is mostly going to be about DNA in one form or another. Then we talked about duplicating chromosomes. We talked about how the fact that a chromosome starts off as one single strand, and then in the S phase of interphase, it duplicates. And at that point, they're attached to each other, right? So you've got these two duplicated chromosomes attached to each other. It's at the centromere. Those things are called sister chromatids. And then once it goes through mitosis, those things separate, at which point they're then considered full-blown chromosomes again. Yeah, and then we have where we are now. Finally, as far as the test is concerned, the most important part of this chapter is the cell cycle, well, and then mitosis, and then meiosis. So, but the point is, we're just now getting into the most important part as far as the exam is concerned. So far, everything we've talked about in this chapter is to prepare you for what we're about to talk about. And the good news is most of you have already done the lab, or at least the pre-lab, so you've already been exposed to this information. So a lot of this is a review. So I can go quickly. But if I go too quickly, obviously slow me down and let me know. The cell cycle, what is it? Again, you've already read it in the introduction in the lab. It is the ordered sequence of events when it comes to your cell. And I'll tell you this for the exam, I'm not going to ask you for the definition of the cell cycle. You're just going to need to know it. You need to know what stage this happens, which stage that happens, what is this a part of, what is that a part of, and we'll come back to that. The cell cycle starts when the first cell is formed until that one cell divides into two new cells. So that is the entirety of the cell cycle. You might consider it the lifetime of a cell, which is a little bit confusing because, you know, we think of things as humans. And for a human, the life cycle is, we consider it from birth to death, right? Or some people might consider it from conception to death, whatever. But here, it's from birth to reproduction. Because in a sense, I don't want to use the word death, but remember, when a cell reproduces, and we have talked about this previously last week, when a cell reproduces, it goes from that one cell, and it kind of splits off into two, right? So it is, in a sense, that is, it's not that the cell necessarily dies. No, it doesn't die. It's just that it splits into two new cells. So it is no longer one cell, it is two new cells. So it is this, the lifetime of a cell cycle isn't from birth to death. It's more like from birth to new birth, if you will. It's not death. Now, of course, cells do die, but that's a whole different discussion. So everything on this slide, I'm going to show you a picture of on the next slide. This is the cell cycle. This is what you need to know. We're going to talk about all these individually. But the biggest mistake, and I'll bring it up now, and I'm going to keep bringing it up as we have this discussion, is that the whole thing is broken down into two main stages, and we're about to talk about it, but still a little preview. There's the big part, which is interphase, right? That's these three things, G1, S, and G2. And then there's this little small part called the mitosis, the mitotic phase. Those are two separate things. The entire life cycle of a cell is broken down into those two separate things. Sort of like a day, right? You might say the day is broken down into daytime and nighttime, the 24-hour period, daytime and nighttime. And from there, you could subcategorize it, right? So, you know, you could have morning and then noon and then afternoon, right? You could subcategorize it. Same here. So overall, it's only interphase and mitotic phase. But then the interphase is subcategorized and the mitotic phase is subcategorized. And that's how that's going to work. Now, there's a lot of information on this slide that I'm skipping over right now because we're going to come back to it. Otherwise, I would ask you, do you have any questions about what's on this slide? But I'm not going to yet because, again, we're about to cover some of this stuff on the slide in more detail. And oh, yeah, let me point this out, too. I want to mention this again because it's been half a week since I mentioned it. A lot of stuff happens in cellular division, cellular reproduction. But what we are focused on in Chapter 8 and in this course is mostly what happens with the chromosomes and, you know, the nucleus, because that's what contains the chromosomes. So you see, that's what we're showing pictures of, is the chromosomes, right? So keep that in mind. All we're talking about for the most part is the chromosomes do this and the chromosomes do that and the cell's doing this in preparation to split. We're talking about this whole thing in the context of the mitotic phase. But keep in the back of your mind, not for the exam or for the semester, because we will never talk about it again, but keep in the back of your mind, a lot of other stuff is happening in this. And one of you, I forget who, I think it was Reed, I'm not sure, but somebody just turned in a good independent work paper about growth and it talks a lot about the different things that cells do.
It was me.
Yeah, that was a good paper. That was a great thing to learn about because, again, all we're focused on is what happens to the chromosomes, but a lot of stuff happens that we don't talk about. So again, now's the part where we're going to get into the detail, and you need to take notes if you're a note taker. Now we're getting into the cell cycle. Interphase, that is this portion, right? That is the non-mitotic phase. That is everything other than the mitotic phase. Interphase accounts for about 90% of the cell cycle. That is a number you do not need to know. Oops, sorry guys. So let me show you by crossing that out. It's not that that isn't true. Again, I'm just crossing it out for your note taking to remind you that that is not a number you need to know. It's the number you don't need to know. What you do need to know is that mostly the cell is in the interphase. Because the mitotic phase, it's just about splitting up, basically. That's its one function. It's like, all right, it's time to split into some new cells. So usually a cell is just doing its own business. And of course, there are exceptions to that. Again, referring back to Reedy's paper, like the paper that Reedy, the paper that you sent, that you referenced. If you read the whole thing in detail, it really talks about like how, depending on what type of cell, it might spend more time in interphase and less time in mitotic phase, or vice versa. But again, we don't get into that. So in the interphase, here's what's happening. Not that you need to write this down. This is for your own information. The cell is just going to perform its normal functions. So if it's a kidney cell, it's going to do its kidney business. If it's a liver cell, it's going to do its liver business. If it's a brain cell, it's doing its brain business. Whatever kind of cell it is, that's just it's just doing what it does in the interphase. It's also going to grow in size, and your book says it's going to double in cytoplasm. Now, this is a little bit contrary to what Hank said in those videos I shared with you. His point was that, you know, when we go through mitosis, which is not what we're talking about currently, but when it does go through mitosis, the fact that you are copying the genetic material means that you don't necessarily need to double everything in the chromosome, because the instructions on how to, I said chromosome, you don't need to necessarily double everything in the cell, because the instructions on how to do that are written in the chromosomes. So again, I'm going to put an X through this. This is just for your own information. I'm not going to ask you, because again, in this chapter, we are mostly focused on what's happening with the chromosomes. I mean, really, in this whole exam, chapters 8, 9, and 10, it's going to be all about what's happening with the DNA. Nonetheless, are there any questions about what's on this side? We're still going to talk about the cell cycle some more, but is there any questions about this particular set of information? All right, like I mentioned, you know, a 24-hour period could be broken down into daytime and nighttime. And even that could be subcategorized, right? The same thing with the cell cycle. The cell cycle overall is broken down into interphase and mitotic phase. And again, overall, we are talking about interphase. But now we are breaking interphase down into its smaller components. And we're not going to do it chronologically, because chronologically we would talk about G1 and then S phase and then G2. But we're not. We're not going to look at it chronologically. We're going to look at it more of relevance. Because as far as relevance is concerned, the S phase is the only thing we're really concerned about interphase, because all we care about in this chapter is what's happening with the chromosomes and the nuclei, or the nucleus. So anyway, let's talk about what happens in the S phase. I just said this, but I'll say it again because it's important. Please don't mess this up. The S phase is part of the interphase. It is not part of the mitotic phase. It is part of the interphase. Or it does not, like, for example, some people think, I've seen this mistake before, it does not go interphase, S phase, mitosis, right? No, it's interphase, the big interphase, and S phase is just a portion of that. So S phase is part of interphase. Here's the other very important part, and this is definitely going to be a test question, and you already had it as a pre-lab question. Chromosomes duplicate in the S phase of interphase. This is when chromosome duplication happens. And again, that is important, the most important thing in the context of this chapter for interphase, because that's all we care about in this chapter for the most part, is what's happening with the chromosomes. And the easy way to remember that is the S for S phase stands for synthesis. I'm not going to ask you what S stands for. That's just a hint. That's just my way of helping you remember when you get the test question, at which stage does DNA duplication happen? It is S phase. And I'm going to put quotation marks around this word because the question, when I ask it, it's not going to be verbatim. I might say chromosome duplication. I might say chromosome replication, something like that. So, sometimes words in biology, it's those exact terms, sometimes not. So I'm letting you know right now, this one is not necessarily duplication. Or I might say, in which stage is the DNA doubled or the chromosomes doubled, So get the big picture, not necessarily that exact terminology. Any questions about S phase? All right, you guys are pretty easy. Like I said, if you've done the lab, especially the pre-lab, this is pretty much a review. This brings us to the gap phases. G1, which comes before S, and then G2, which comes after S. Let me put a big old X through this before we even talk about it. Meaning, again, for the exam, you don't need to know any of this. If anything, just know the sequence. Just know first comes G1, then S, then G2. That's it. Know what order it's in. And that's a big maybe. I might not. If that question's on there, it'll probably be extra credit. But nonetheless, your book talks about it, and it is good to know these things, so let's talk about it. What happens in G1? Well, again, like I said earlier, G1 phase comes before the S phase. In this situation, actually, you know what? I take that back. Again, even though I'm not going to ask you these questions, I can't undo it easily. Anyway, even though I'm not going to ask you questions about this, all the information on here is something you should know. I know that sounds weird, but let me explain. You do need to know what happens in S phase, right? You do need to know that S phase is when the chromosomes duplicate. Therefore, logically, you should know that before S phase, the chromosomes have not duplicated. Therefore, in G1, which is before S phase, the chromosomes are single, right? That's the important part, right? The chromosomes are just single chromosomes. They have not duplicated yet. That would be the important part. And then the less important part in the context of this chapter and for this exam would be that the cells are just performing normal functions. Your kidney cells are doing kidney things. Your eyeball cells are doing eyeball things. Your skin cells are doing skin cell things, whatever it is. They're just living their best life. Then, after S phase comes G2, the second gap. And again, even though I'm not going to ask you this, you should know that in the S phase, the chromosomes have doubled. Therefore, the phase after that, which is G2, the chromosomes are now doubled. They consist of 2 sister chromatids. You should know that, even though I'm not going to ask. That just logically makes sense that the stage after the duplication stage, you would have 2 sister chromatids as opposed to that one single chromosome. And of course, your book points out also that at this point, your cell is preparing to divide. So whatever kind of cell it is, actually, I should say, depending on what kind of cell it is, it could be doing like nothing but preparing to divide, or maybe it's still doing its normal function a little bit, but doing a lot more preparing to divide, or maybe just, doing a lot of its normal function and a little bit preparing to divide. It depends on the type of cell. But the point here is, again, for the exam, you need to know that G1, you have a single chromosome. G2, you have the sister chromatids. Because, more importantly, S phase is when the chromosomes duplicate. And S phase is the thing between G1 and G2. So any questions about interphase in general? Okay. Notice again, interphase is not a part of the mitotic phase. And this confused me as a freshman biology student because no one pointed it out a lot, and the words just blended together. So I want to make sure they're not blending together. Every time we talk about mitosis, we do have a conversation about interphase, right? You can't have mitosis without interphase. So those questions, those words always blend together. But remember, interphase is not part of mitosis. Some of you got that wrong on the study guide, and I don't blame you. It's an easy mistake to make because not the study guide, excuse me, the pre-lab. It says list the phases of mitosis in order, and some people slightly incorrectly put interphase, prophase, metaphase, anaphase, telophase. And again, that is wrong because interphase is not technically part of mitosis. I mean, you are correct when you say that interphase comes before metaphase or prophase. That is true. Interphase does become before prophase, but interphase is not technically part of mitosis. And I just wanted to make that clear because, again, as a student, that kind of confused me at first. Anyway, any questions about that? All right. You guys must be sleepy. This is sleepy weather. All right, the next word for attendance, I'm going to underline. I'm not going to say it, just to make sure everybody online is actually paying attention with their eyeballs and their ears. But that is the next word for attendance. Anyway, we are done talking about the interphase. Now we're talking about, as far as the exam is concerned, the more important part, which is mitosis. We're going to spend more time. You will be spent more time getting tested on mitosis than you will interphase. Again, I say this again because I want to make sure it's clear because it's easy to mess up. Mitosis is not a part of interphase. If the whole cell cycle is broken down into two parts, which it is, one part is interphase and the other part is the mitotic phase. So here's where it gets a little bit more confusing. And if this confuses you, don't worry about it too much because there's probably not going to be any test questions on it. But the same way interphase is broken down into G1, S, and G2, the mitotic phase is also broken down into two separate parts, which is mitosis and cytokinesis. And I say that's confusing because generally speaking, we talk about these things together again. Those words blend together the same way interphase seems to be like a part that comes right before prophase, and it is. But remember, it's not technically part of the mitotic phase. The way that so to me, this can be a little bit confusing too because we're breaking down the mitotic phase into two separate phases, mitosis and cytokinesis. But the reason this is trickier is because cytokinesis, it's not distinct. Like if you look here, you go, bam, from G1 to S, and from S to G2, right? It's distinct. The mitosis and cytokinesis, mitosis is happening, like that's a big chunk, and then just a part of that at the end is also cytokinesis. So that's why I don't like, for the exam, I'm not necessarily going to ask you to differentiate between the two. You need to know what cytokinesis is, but in your mind, you can just think of cytokinesis as the end of mitosis. Now, whether you recognize that as an overlapping part of mitosis or not, I don't care about that. Just know that that comes at the end. Anyway, that being said, what happens in mitosis? You don't need to write this down because you're going to need to know the subcategories of mitosis. So this is just a preview. But in mitosis, the nucleus and its contents divide into two daughter nuclei. So again, if you're writing things down, there's no need to take notes on mitosis yet because we're going to dive deeper into that. Actually, the same can be said for cytokinesis. But what happens in cytokinesis? That is when the cytoplasm, which is like the cell itself, like not just the nucleus, but everything else, and all of its organelles, it divides into two. So again, mitosis, that's strictly looking at the nucleus and everything inside of it. That's what we're talking about. Mitosis is dividing the nucleus, right? Cytokinesis, everything else. That's how you can look at it. Mitosis, nucleus, and everything in it. Cytokinesis, everything else. There's a question on your study guide that might be on the exam, and it's a great question. It's a great way to think about things. What would happen if a cell were to complete mitosis but not cytokinesis? And there's a lot of options on there, but let's just talk about the correct option. If a cell were to go through mitosis, which divides the nucleus, but not mitosis, which cytokinesis, which divides everything else, then what you'd be left with, well, let me ask you, how many cells would you be left with? Let me just ask you guys. If it went through mitosis, but not cytokinesis, now that you have this information right here in front of you, if you went through mitosis, but not cytokinesis, how many cells would you have? You started with one cell, you went through mitosis.
Just two? Oh, no, wait.
Someone said it. You only have one, right?
Oh,
because in mitosis, you're only splitting the chromosome, the nucleus and everything inside of it. Cytokinesis is the thing that actually splits the cell. So at the end of mitosis, if you didn't go through cytokinesis, you would only have one cell, but you would have two nuclei. Not that that happens very often, but that's just a good, I like that question, not because it's important, because it happens a lot in your everyday life, or that it's important for understanding things later in this semester. I like it because it requires you to think instead of just, memorizing things that are on the book and then spitting them out. It makes you think like, all right, if this thing went right and this thing went wrong, and I know this produces that and that produces this, then what's the results, right? I love those kind of questions, but it's a 100 level class, so you don't get a lot of those kind of questions. Anyway, so the combination of mitosis and cytokinesis give us this: 2 genetically identical daughter cells. You're going to see that a lot, so I'll underline it again because we are talking about mitosis. Mitosis and cytokinesis. The result of mitosis and cytokinesis are 2 genetically identical daughter cells. The key there is 2 and genetically identical, because a lot of the questions, or some of the questions on the exam, are going to be you comparing and contrasting the difference between mitosis and meiosis. And only mitosis gives you 2 cells instead of 4. And only mitosis gives you genetically identical cells as opposed to genetically unique. And also, that's a good pre-lab test or pre-lab question that you've probably already done. Hopefully, you've already done it. And if not, this is one of the reasons you get half credit for turning it in late, because very often I give you the answers. So anyway, any questions about this slide? Okay, let's get into it. Let's actually dig into the different stages of mitosis and cytokinesis. And again, I recommend watching that video that I posted, that little short video with Hank. He explains it quickly, he explains it clearly, and he does it in a more entertaining fashion. But let's get into it. Oh yeah, let me say this too before we get into it. Because here you can see this. You are getting the Cliff Notes version. You are getting the non-biology majors 100 level version of mitosis. It is actually a little bit more difficult, a little bit more complicated what I'm giving you. And I saw this mistake on the pre-lab, so it's a good time to bring this up now. When it comes to exams and what you're tested on, you are responsible for the information in your textbook. So if you find something else outside of the textbook, that's even great. That's great. You can write about it in an independent work. Of course, it's a multiple choice exam, so you shouldn't mess this up. But for example, there is something called Metaphase that we do not talk about in this course. And it was not on your lab introduction. Nowhere in the lab did it say anything about Metaphase. But people found the answer. And I'm not like talking bad, because that's great that you went out and found more information. Good on you. I'm just saying, as far as grading is concerned, always go back to what you're responsible for. Or I think some people, sorry, I'm getting a little sidetracked here. I still think, even though we're halfway through the semester, that some people don't know all the information you need for your pre-lab is on the lab. Unless otherwise stated, there's never any reason for you to read anything else or watch anything else. It should all be there on the lab, just to make things easier for you. But again, if you want to look out and find more information, that's even better. The more the merrier when it comes to knowledge. Anyway, here we go. Let's get into it. Mitosis is broken down into 4 phases. There's no need to write this down yet, because we're going to talk about them individually. But this is them in sequence: prophase, metaphase, anaphase, telophase. There will be a test question that asks you to, which of the 4 or which of the following are the phases of mitosis in the correct order. And the answer is prophase, metaphase, anaphase, telophase. Most people will get that right. Then there's going to be a test question that looks very similar that says something different, like, in which phase are the chromosomes meeting in the middle? And it'll be a blank. And in this phase, the chromosomes are completely separated and on different sides of the cell. And in this phase, the chromosomes have coiled up. And in this phase, the chromosomes, sister chromatids, have just split up. It'll say something like that. It'll be blank, 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 blank. And most of you guys will get it wrong, even though I'm telling you right now, that is not the same question as put them in order. There's a different question that says, in this stage, this happens, in this stage, that happens. So don't confuse the two. I'm hoping you won't. Maybe you'll be the first semester where you actually follow this advice and pay attention and don't miss that question. But yes, there's 2 separate questions. Anyway, let's talk about what happens. And again, what we're focused on mostly in this discussion is the chromosomes. So just keep in mind that there's a lot of other things happening that we just don't talk about. I'm giving you the simple Cliff Notes version. All right, the first thing you need to know before we even get into the different stages is, again, the whole thing is about chromosomes moving, right? Where are the chromosomes going? Where do they come from? Where do they go? Blah, blah, blah. We're going to talk about all that. So then if we're going to talk about them moving, you have to wonder, well, how are they moving? What's making them move? And there's something called the mitotic spindle. I'm probably not going to directly ask you a question about the mitotic spindle. I don't think, or maybe I will for extra credit. I don't know yet. But I am going to use the word a lot. Well, maybe not a lot, but I will use it. So you need to know what I'm saying when I say it. Your book describes the mitotic spindle as a football-shaped structure of microtubules. And as far as you're concerned, microtubules are just things that are attached to the chromosomes. They do other things too, but in this class, we won't even talk about it. Microtubules are just things that attach to the chromosomes and move them along. You can almost think of them as ropes. As far as we're concerned, they are ropes. They're just ropes that attach to the chromosomes that move them around. So that is what a mitotic spindle is. And again, I'm probably not going to ask you about it, but you need to know what it is because I'm going to be using the word. So here's a picture. Well, this is metaphase, not that you need to know, but see these things? Well, we won't even talk about that yet. But those things, these little rope-looking things sticking out, attached to the chromosomes, those are the mitotic spindles. Those things are literally moving the chromosomes around. And again, the story, especially for the exam, the story of mitosis is going to be, where are the chromosomes? Like their movement, right? Where are they moving to? Where have they just come from? So keep that in mind. Any questions about this slide? Okay. The next attendance word will be neon. You know, like that element, or like a sign that glows, or like that song by John Mayer. Probably other songs too called neon, but oh yeah, what's that? Brooks and Dunn, neon. I can't think of it right now. Doesn't matter. It's not a music class. Anyway, are there any questions about this slide? All right, let's move forward. So again, the whole thing here, this whole story, as far as we're concerned about mitosis, is about moving chromosomes. Specifically, we've got one bag of chromosomes. Let me get the screen to see me. We've got one bag of chromosomes, if you will, which is the nucleus. That's what we start with. And what we want to end with is two bags of chromosomes, right? Two nuclei. Now, when they start off, they're not condensed, right? They're all out there. So let me ask you this. I wish I had a bag with me. I don't. If I had a bag of yarn, hold on, I want to see if I want to start that off yet. Let me ask you this. Okay. We have Ramses here, right? You guys can't see online, but we have Ramses on one side. We've got White on the other, right? Now imagine if I was like, all right, guys, we're gonna have a race. And I don't know if you guys can see the table online, but let's say I had two of these tables and White got this table and one of the Ramses, maybe you got that table. And I said, all right, here's what you need to do. I need you to take this yarn, these 9 balls of yarn, and I want you to segregate it to where, well, it's not like 9 because you can't do that in half. But anyway, half the balls, half the yarn on one side and half the yarn on the other, right? Let's say that was the race. And this is what White had. And this is what the Ramses had. Who do you think is going to win the race? Splitting those balls of yarn from one side to the other, or splitting that yarn up even. Yeah, probably. And if you can't hear online, one of the Ramses probably said not us, and they're probably correct. Probably this one would be easier to split, right? It's just them balls. All you have to do is be like, okay, some balls here, some balls here. The Ramses got this big old tangled mess that they'd have to deal with. Does that make sense? So in order to efficiently move this yarn from one side to the other, it would be best if it was in balls, right? Not this big, stringy, tangled mess. All right, now let me ask you this, and I wish I had a bag. I don't. Let's say again, I have Ramsey, the other Ramsey this time. So the other two raced, White won. The other Ramsey, I give you this ball of these balls of yarn, and I give it to you in a Kroger bag. All right. I need you to do the same thing. It's not a race. I just need you to take these yarns, this yarn, and separate it half and half. What's the first thing that she would do if I gave her that ball of yarn? That Kroger bag full of yarn, and I told her to separate it on the table. What's the first thing you would do? Take it out of the bag, right? All right, so keep that in mind. We know logically that to separate this yarn, first thing you need to do is take it out of the bag, because if it's in the bag, you can't get it on separate ends of the table. And second, we know that it would make a lot more sense if it was all balled up as opposed to a big squiggly mess. Keep that in mind when we talk about what we're about to talk about, which is the first phase of mitosis. Finally, we're here. It is called prophase. Pro, like a professional athlete. We're number one. We're number one. Number one. Number one. Number one. Remember that. Pro, number one. Anyway, I will even write that. Number one. So let's talk about what happens in prophase. And remember, this is the multiple choice exam for the most part. This part will probably be fill in the blank. So there will be no question where I say, tell me everything that happened. Well, unless you have the harder version because you took it late, or maybe this will be an extra credit question. But generally speaking, you're not going to have a question where I say, all right, tell me everything that happens in prophase. More likely what will happen is I'll tell you some of the stuff that happens in prophase, or all of it, and you just have to say, oh yeah, I recognize that stuff. That's prophase. You see what I mean? It's one way, not the other. It'll be me telling you the stuff and then you recognizing it as prophase, as opposed to me saying, hey, tell me everything that happens in prophase. Anyway, this is not in chronological order. This is just the order in which we are talking about it. But in prophase, the chromosomes coil up. Because again, when they're not going through mitosis, it's a big squiggly mess. And if you've done the lab already, you've seen, like if you look at these microscope pictures, you can't tell one chromosome from the other. You can't even tell any chromosome. It just looks like a big, like cloudy mess in that picture because the chromosomes are not coiled up. They need to be uncoiled, and you'll learn why later in the semester. But basically, there's cellular machinery that needs to clamp onto that DNA and read the code. And if it's all coiled up, it's going to be a little bit harder to do that. But anyway, the most important thing you need to know for the exam is that the chromosomes coil. And remember the balls of yarn to help you remember that. If you're going to move a bunch of yarn from one side to the other, you need to make little balls of yarn. Same thing with the chromosomes. If you're going to move the chromosomes from one side of the cell to the other, it would be easier if they coiled up. At this point, even though this is not new information, we already talked about this when we talked about the S phase of interphase. At this point, these chromosomes that have coiled up, they still exist as sister chromatids joined at the centromere, which would be this picture here. I mean, it's just a meme, but that's what that is, right? Those are sister chromatids, right? That's why it has that X shape. This is one chromosome, and this is the other one, which has been duplicated, and they're joined together right there at the centromere. Those are sister chromatids. Even though the test question won't go like this, that could be one of the tests. It could be, if I were to write it that way, I could say, which of the four phases of mitosis, in which of the four phases of mitosis are the sister chromatids still together? And prophase would be one of the answers to that, because they are still together. And that's not always the case in this situation, in mitosis. All right, next bullet point. The nuclear envelope has to break into pieces. And we already had this discussion when we talked about the yarn in the Kroger bag. If I were to ask the other Ramsey to separate the yarn, on this table, I don't know if they can see it or not, but half of it here, and I gave her a bag of yarn, the first thing she's gonna have to do is take it out of the bag. Same thing with the chromosomes. If you're gonna separate them, if we go to one side of the cell and the other, but they're in a bag, aka the nucleus, then you've got to get rid of the nucleus, and that's what happens. It breaks into pieces. I should have thought about this earlier. Let me put that. This is not one of those verbatim things. I might say breaks into pieces. I might say dissolves. I might say disintegrates. I might say disappears. The point is the nuclear envelope goes away. So it's not verbatim, breaks into pieces. Unlike this bullet point that we just finished, the chromosomes do exist verbatim as sister chromatids joined at the centromere. Those are the exact words. We're not using other words for that. But again, the nuclear envelope disappears, goes away, disintegrates. Next, that mitotic spindle that we talked about that moves the chromosomes around, it attaches to the centromeres. That's this. And I'll even put that in quotation marks because I might say spindle, I might say mitotic spindle, I might say attaches, I might say connects. I might say centromeres, and again, here on the right, that's the centromere, right? So I could also just say, instead of being so specific and saying they attach to the centromeres, I could say they attach to the chromosomes, because it's the same thing. I'm just being more specific when I say the centromeres, because that's specifically where they attach. So again, for this bullet point, don't memorize the words verbatim. Just understand the thing that moves the chromosomes around, it attaches to the chromosomes. And that makes sense, because if this whole thing is all about moving chromosomes around, logically the first step needs to be, or one of the things that happens in the first step has to be the thing that moves them around attaches to them. Does that make sense? Logically, that has to be part of the first step. And then finally, the last part, and this is the part that's not as logical. Oops. This last bullet point is less logical, less intuitive, I guess, but still important. This is when the chromosomes start to move towards the center of the cell. So again, the centromeres attach, or excuse me, the spindles attach and start moving them. Where do they start moving? They start moving them towards the cell. So you need to know all this for the exam. Again, not because I'm going to ask you to list it, but because I'm going to give you all or some of this, and you need to recognize that as prophase. And I hope this is all common sense. Not common sense, but I'm hoping this is all logical to you. Like I said, all of this has to make sense. The chromosomes have to coil up before you move them. You saw the yarn. That's logical. That has to happen. I guess that part's not as logical. That's just some words, some terminology. But the nuclear envelope disappearing, that has to happen, right? You can't separate them if they're stuck in a bag together. The spindle attaching, the thing that's moving them, obviously that has to happen. You can't move them around until the thing that moves them around attaches to them. So yeah, all that I'm hoping is very logical. And then again, the rest of the prophase, metaphase, anaphase, telophase, it's going to be less detailed, and it's going to be more about what's happening to the chromosomes, and that's where this bullet point comes in. And which of the four phases are the chromosomes moving towards the center of the cell? And the answer is prophase. In prophase, the chromosomes are moving towards the center of the cell. So let's focus on that for a second. And we don't do this, but if we are defining prophase by the fact that chromosomes are moving towards the center of the cell, then what do you think the next phase might be? Forget the name of it, but the action. What do you think defines the next phase? If this phase is the chromosomes moving towards the center of the cell, what do you think the next phase is? Not the name, but what do you think happens that says, okay, this is the next phase? And just read that last bullet point and think about it. Perfect. Logically, right? That has to be it. If this is defined by the fact that the chromosomes are moving towards the center of the cell, then logically the next phase must be they made it. And that is it. Well, excuse me. First, let me show you some pictures. Again, here's interphase. Like we said, the chromosomes are uncoiled. You can't tell one chromosome from the next. The centromeres have not even made the centrosomes, which we haven't even talked about. They haven't made the spindle. The spindle hasn't attached. That's just regular old interphase business. Then we get into what we're talking about now, which is prophase. The chromosomes coil up because, again, if we're going to separate the yarn, we want balls of yarn. The mitotic spindle starts forming because that's the thing that moves the chromosomes, and this is all about moving chromosomes. Then the nuclear envelope disappears because, again, you need to get rid of the bag if you're going to move the yarn. So it's gone. The centromeres attach because that's the thing that moves the chromosomes. And again, at this point in prophase, the chromosomes are moving towards the middle of the cell. They're not there. They're moving in that direction. And like one of the Ramsey said, the next phase is just that. So I'll talk about that one first. I'll go out of order. In metaphase, we could say it is defined by the fact that the centromeres are lined up between the two poles of the spindle. I'm going to put quotations marks around here too, because again, this isn't a verbatim thing. So again, in prophase, the chromosomes are going towards the center of the cell. In metaphase, they're there. The chromosomes are lined up in the middle of the cell. And notice I said it different that time, right? The way this is written, that bullet point is directly from your textbook. Centromeres are lined up between the two poles of the spindle. But again, I like the terminology. The chromosomes are lined up in the middle of the cell. So again, that's why I put those quotation marks around it. Don't memorize that verbatim. Just know that this is the point. where the chromosomes made it. Boom, they made it to the middle because that's what they were doing in prophase. They were headed to the center. They were headed to the middle. And now they made it. I'm going to put an X through this one. This came from your book. But yes, the spindle is fully formed. I shouldn't have even included that now that I'm thinking about it. Of course it's fully formed. Because like we said, I don't want to get into the details. But the way we're telling the story, it would make sense that it's fully formed because I already told you that the chromosomes were on their way towards the center in the last one, right? And the spindle is the thing that moves them. Now, that is technically a correct bullet point, but we don't get into the details of that. So again, the most important thing as far as the exam is concerned, in metaphase, that is this picture right here. The chromosomes are lined up in the middle of the cell. And the way I remembered it as a student, again, prophase, professional athlete, pro, number one, right? That's number one. Pro is number one. And then what happens in metaphase, I always remembered, okay, that's when all the chromosomes met in the middle, if that helps you. That's not at all what it means with the Greek prefixes. You can look that up if you want, but either way, it helped me remember it. That's where they met in the middle. Anyway, at this point, here they are all in the middle. And then look at this bullet point right here. They're not just sitting there. What's happening now is these spindles are pulling. See, if you look on the right, I'm drawing arrows. They're pulling in opposite directions. It's like a tug of war with the chromosomes, or excuse me, the sister chromatids in the middle, right? They're pulling, they're pulling, they're pulling. That's what's happening right now. As a matter of fact, that's what's keeping them in the middle, is because it's a tug of war and nobody is winning yet, right? That's why they're in the middle. So again, we kind of define the beginning of metaphase as, even though we kind of define metaphase as the chromosomes are in the middle, right? Now that being said, like I said, now that they're in the middle, they are being pulled. We can't see it, but it's being pulled, right? They're being pulled. So logically, if they're being pulled right now, what must the next step be? Not the name of it, but if this phase is they're all in the middle getting pulled, then what must the next phase be? What happens in the next phase? And let me back up to remind you, because somebody got this last one. In prophase, I said, all right, the chromosomes are moving towards the center. So what is the next phase? And then one of the Ramseys correctly said, well, if the prophase is then moving towards the center of the cell, then them getting to the center of the cell must be metaphase. And that was correct. And now I'm telling you, here in metaphase, they're sitting there getting pulled, right? They're getting pulled apart, but they're still together. So what must the next phase be? If right now they're getting pulled in opposite directions, what must the next phase be? Don't overthink it. Think. Is that the same Ramsey again? All right. Remind me of this. I'm going to give you some extra credit because you keep getting them right. Yes, so you didn't hear what she said. What she says, they are pulled apart, right? So in metaphase, they're getting pulled, right? And then in the next phase, which is anaphase, that's it. They finally have been pulled apart. Excuse me. Another way of saying that is the sister chromatids have separated. And again, I'm going to put that in quotation marks because it's not one of these verbatim things. It's not the exact verbiage that I'm using. It's the fact that in that last phase, they were all lined up in the middle, getting yanked and pulled, and then finally, boom, they break. And once they do, that is what we call anaphase. And this is a good point to remind you that, again, you are getting the 101 version, the easy version. Because think about it, it's not all happening at once. Like, it's not like they all split at once. So really, the transition from one phase to the next is sometimes a little bit sloppy. It's not necessarily distinct, just to keep in the back of your mind. But for the purposes of this 100 level class, we just talk about it like first it's prophase and then it's metaphase, right? Very strict, but usually it's really kind of blends in from one phase to the next. Anyway, at this point, once those sister chromatids are separated, like you can see in this picture on the right, they are no longer considered sister chromatids, right? At that point, they are now considered full-fledged chromosomes. And again, a test question I mentioned earlier that I might ask, but probably won't. I never have before, but why not? Which of the phases of mitosis are you dealing with sister chromatids, right? And that would be the first two. And then if I said, in which of the stages of mitosis are you dealing with full-fledged chromosomes? That would be anaphase, and of course, the one we're going to talk about next. The next word for attendance will be sand. Like the things, all that stuff at the beach, sand. Anyway, obviously the chromosomes were getting pulled because that's what kind of defined anaphase, right? Was they finally got separated. Now we know they are moving towards the other end of the pool. It's not like they get separated and this is a video and they got separated and they're like, all right, cool, we're done. And they just sit here. No, what's happening is these things are still moving in this direction. They're still moving in the other direction. And also, mind you, another thing you can't see is that this round thing here is going, it's getting more of an oval shape because this is also moving outwards, right? And this is kind of moving inwards relatively. So it's getting elongated. This part, I'm going to put an X through because you don't need to know that for the exam. That's just for your own knowledge. Just imagine this thing going from a circle to an oval. Anyway, in anaphase, all right, I'm going to finish this. If in anaphase, if the chromosomes are moving in separate directions towards the end of the other cell, then what must the next phase be? Not by name, but how would we define the next phase? If in this phase they're moving towards opposite sides, then what's the next phase? Come on, guys. We don't have time. They made it, right? So if in this phase they're going towards the opposite ends, in the next phase they did it. They are now at opposite ends. So that's it for the day. I'll see you online for lab if you haven't had your lab graded by then, and I will be online for office hours. Sorry. Well, we made it in time. No, we're 1 minute late. So I apologize for keeping you 1 minute late.
Have a good one.
You too. And remember, you don't have to do the lab alone. Between now and then, if you need my help, let me know. I will help you.